Sutro's innovative technology has lended to 5 clinical programs. Our third wholly owned ADC program, STRO-003, is an ADC that targets the tumor antigen ROR1 and uses a novel linker payload, which is an exatecan DNA targeting mechanism to kill the tumor. STRO-003 is very exciting because ROR1 is expressed in diverse cancers, both blood cancers as well as many solid tumors, including non-small cell lung and triple-negative breast cancer. We've gone after ROR1 by using this new novel linker payload, targeting DNA within those tumor cells, which has been shown clinically to be a very effective and potent way to kill the tumor. We've also combined these novel and potent exatecans with a new linker system, which gives us very good stability and good tolerability in all of our non-clinical models. We've been able to conjugate 8 linker payloads onto a single antibody. Going with this high DAR in this site-specific manner really allows us to get as much activity for each binding and internalization event. What this means is that every drug, every molecule is really optimized to really harness as much activity against that tumor as we can. So we're really excited that we're going to be seeing STRO-003, this new ROR1 ADC, in IND-enabling studies and approaching the clinic to really help to address a lot of unmet need, both in hematology as well as solid tumors.